Hello viewers. In last video about antibody structure, we learned about the basic structure of antibodies, concepts like how many chains, how many polypeptide chains these are made up of, like heavy and light chains, and what kind of domains they have, like immunoglobulin domain, and whether they are glycoproteins or simple proteins. Okay, all those things we learned in our previous lecture. Now in this lecture, I would focus on digestion products of antibodies. So digestion means cleavage or digestion of polypeptide backbone, right? Or we can say peptide bond. So peptide bonds are can be digested using proteases, right? So papain digestion of an IgG molecule has been taken here as an example. For the first time, it was done by Porter in 1950s. Papain, which is a protease, cleaves an antibody molecule just above the disulfide bonds. The light and heavy chains are held together by disulfide bonds, right? And both the heavy chains are also held together at hinge region or at CH2 just before CH2 position. At hinge region, interchain disulfide bonds are there, and this papain enzyme cleaves these antibody molecules just above the interchain disulfide bonds between the heavy chains, right? And when you run the digested products, you will find only two bands corresponding to 45 kilodalton molecular weight and 50 kilodalton molecular weight. Now, when certain other studies were carried out to check which fragment with either 45 kilodalton or 50 kilodalton interacts with the antigens, it was found that only the fragment that is of 45 kilodalton can interact with the antigen, not the fragment having the 50 kilodalton weight, right? So this fragment of 45 kilodalton was named as fragment that is capable of binding to the antigen. So fragment antigen binding. So although there are two fragments, but because of their equivalence in weight, we get only one band. And the rest, the single fragment that we are getting of 50 kilodalton is known as fragment that is crystallizable, FC. FAB and FC. So FAB for fragment antigen binding and FC for fragment crystallizable. Because crystallization of this is very easy. High concentration during the storage get automatically crystallized. If we talk about pepsin digestion of an IgG molecule, which was for the first time carried out by Nisonoff in 1950s, what happens during this pepsin digestion of an antibody molecule? Uh, this pepsin digestion occurs just below the disulfide bonds, right? So there is no chance of separation of these two heavy chain products, right? And rest of the heavy chain gets digested completely. So when you run the products of this proteolytic digestion, you only get 100 kilodalton protein. Okay, so this 100 kilodalton protein, now the antigen binding capacity was checked. So it was found that yes, it is also able to interact with the antigen, and hence it was named as fragment antigen binding. But because two proteins are there, or we can say two FABs are there, so this 2 was also written here. In earlier digestion with papain, we only get this much FAB. This is a single. This corresponds to the single FAB. This corresponds to another FAB. So FAB prime and 2. So this is the symbol used for this kind of a digestion product. And because this FC fragment was chewed down, so you get multiple fragments after digestion of this FC. Now, these are the that about four immunoglobulin domains will be there in this FAB2, right? FAB prime 2 fragment. This light chain is having two immunoglobulin domains, two on this light chain and two on each of these heavy chain fragments. right? All of these are held together by disulfide bonds. And I told you in our previous lecture on antibodies that the N-terminus site of the antibody molecule has a variable region, okay, which is having the paratopes that binds to the epitopes. In both the cases, after papain and pepsin digestion, these FAB fragments get that portion, right, variable region. So that's why both of these are able to bind to antibodies, antigens. Now, what will happen if you treat these antibody molecules with a reducing agent like mercaptoethanol? You know, reducing agents are known to reduce disulfide bonds, right? And this thiol group is created or restored again. So when you treat this protein, antibody protein, that is IgG molecule, with mercaptoethanol, these disulfide bonds, interchain disulfide bonds, will break open, right? And these will be converted into SH groups. Now, when you run the reduction products on the gel, you get two protein bands, one corresponding to 50 kilodalton and another 25 kilodalton. As you can clearly see that there are four polypeptides, right? And two are of equal size, rest two are of also equal size. So you can imagine that the heavier one, that is of 50 kilodalton, will be our long chain or heavy chain, and the shorter one of 25 kilodalton is the light chain, right? So you have seen that papain digestion give you the FAB fragment and FC fragment, while pepsin digestion gives you FAB prime 2 fragment of higher weight, that is 100 kilodalton, and mercaptoethanol reduction gives you two proteins, one of 50 kilodalton and another is of 25 kilodalton. Now another concept is that these antibody molecules can itself act as an antigen. If you immunize an organism with antibodies, then antibodies will be raised against these antibodies, which are actually acting as antigens, right? So antibodies raised against a particular fragment, whether it will be able to react with the light chain or heavy chain, it you can easily now say whether these antibodies will bind to heavy or light chain or not. As you know that an FAB fragment is made up of both portion of the heavy chain as well as complete light chain. So an antibody raised against an FAB fragment will be able to bind or react with both heavy as well as light chain. Whereas if you raise an antibody against FC region, which has only the portion of the constant region of the heavy chain, the antibodies raised against this region will only be able to bind to or interact with only heavy chain, right? So as you know that FAB consists of portions of both heavy and light chain, while FC contains only a portion of heavy chain. So now with this video you learnt about the digestion of an antibody molecule with papain, pepsin and reduction with mercaptoethanol, and you also learned different products produced by these digestion as well as reductions. And you also know now that if you raise an antibody molecules against an FAB fragment or FC fragment, whether it will bind to HL, both the chains, or H only, right? So if you want to know more about antibodies, please check out our next video. Thanks for watching and keep coming onto this channel for more videos and more updates. Please do subscribe, like and share. Thank you.